Welcome back to the deep dive. Today, we've got this stack of, well, pretty intense academic sources, the kind you'd usually find in a lecture hall.
Yep. We're cracking them open.
And our focus is super fundamental. Chromosomes, the blueprint of life. And crucially, what happens when that blueprint gets a bit smudged?
Exactly. Our mission, really, is to give you a path through human cytogenetics. We're starting small at the level of cell division mechanics, especially where things can go wrong.
Uh-huh.
And we'll end up with the big picture. The universal language, the codes doctors use everywhere to describe these conditions.
Okay, and just setting the scene, our sources kick off with a mention of a specific genetic case. It's a 14-year-old with an ADA2 mutation, leading to vasculitis, stroke.
Right, and that's a stark reminder, isn't it?
Yeah.
Even tiny single gene changes can cause massive problems throughout the body.
Definitely.
But today, we're kind of zooming out from those individual letters of the DNA code. We're looking at the bigger structures, the chromosomes themselves. Numerical problems, too many or too few. Structural problems, bits missing or rearranged.
So if DNA is the instruction manual,
Yeah,
we're looking at what happens when whole chapters are, duplicated or just ripped out.
precisely. And to get why errors happen, you first have to understand the normal process, the foundation.
Makes sense. So let's start there. The standard way cells copy themselves, mitosis. This keeps our bodies ticking over.
Right, the cell cycle. Most of it, like 90%, is interphase. That's G1, S phase, where DNA copies, G2, growth, prep work.
And the M phase, mitosis itself, is quick.
Relatively quick, yeah. The whole point is genomic stability. You take one cell, you make two genetically identical diploid cells. Your basic body cells, somatic cells. Predictable, stable.
But stability isn't always the goal, is it? Especially for reproduction.
Exactly the opposite, in fact. For the species to thrive, we need diversity, variety. And that's where meiosis comes in. It only happens in germ cells, sperm and egg precursors.
So mitosis stability, meiosis eats diversity.
You got it. With meiosis, one diploid cell ends up producing 4 genetically unique haploid gametes.
Okay, so how does it generate that diversity? What's different?
Key difference. Still one round of DNA replication, just like mitosis. But then it goes through 2 rounds of cell division, meiosis the first and meiosis the second.
2 divisions, and meiosis the first is the crucial one.
That's the big one. It's the reduction division. You go from diploid, 2-inane, having 2 copies of each chromosome, down to haploid, nan having just 1 copy. It's also the high-risk phase.
High risk. Okay. What happens in meiosis? That's so important.
Well, first, in prophase I, something amazing happens. Crossing over. Homologous chromosomes, the pair you got. One from each parent, they physically pair up and swap chunks of DNA.
They actually exchange segments?
Yeah, physically swap bits. Then, in metaphase I, these paired homologous chromosomes line up at the cell's center. And how they line up is random, independent assortment.
Okay, random lineup, DNA swapping. How much variety does that actually create?
A staggering amount. Just from the independent assortment of our 23 pairs of chromosomes, you're looking at over 8 million possible combinations. That's 2 to the power of 23.
8 million. Before you even count the crossing over.
Before you even count crossing over. And crossing over happening at these points called chiasmata adds way more variety. Plus, it's actually mechanically important. It helps hold the pairs together properly so they separate correctly later. It reduces the risk of errors.
Interesting. So nature builds in this complex system for diversity, and crossing over acts like a safety check within that system.
It does. But sometimes that check fails. The system breaks down.
Which brings us to the errors.
Exactly. Non-disjunction. That's the term for it.
Non-disjunction.
It just means the failure of chromosome pairs, or later the sister chromatids, to separate properly during meiosis. Segregation goes wrong.
And the result.
Gametes, sperm, or eggs with the wrong number of chromosomes. Too many or too few.
And I remember reading in the sources, it really matters when this failure happens, right?
Yeah.
Meiosis I versus meiosis the second.
Huge difference. If non-disjunction happens in meiosis the first, the homologous chromosomes fail to separate. That means all 4 resulting gametes are abnormal. 2 get an extra chromosome, N+1. 2 are missing 1, N1.
Okay, so a meiosis I error affects everything downstream. What about meiosis the second?
If the error's in meiosis the second, it's the sister chromatids that fail to split. It's still bad, but less widespread. You end up with 1 gamete that's N+1, 1 that's N1, but also 2 that are perfectly normal N.
Right. And clinically, this happens more often in egg formation, maternal meiosis.
Significantly more often, yes, particularly meiosis I errors.
Why is that? Why are eggs more prone to this?
It really comes down to time. Oocytes, the egg precursor cells, actually start meiosis I before a female is even born. Then they pause, arrested in prophoresi for decades.
Decades, just sitting there.
Yep, waiting for ovulation, maybe 15, 25, 35 years later. And over that time, the protein structures holding those paired chromosomes together, the cohesins, they can start to degrade, break down.
Ah, so it's an age thing. The machinery gets older.
That's the leading theory. This age-related breakdown is why the risk of conditions like Down syndrome, mostly caused by meiosis-I non-disjunction, goes up so dramatically for mothers over 35.
Okay, so non-disjunction leads to abnormal gametes. When one of those meets a normal gamete at
The
fertilization,
result is an embryo with aneuploidy.
aneuploidy, let's define that clearly. How's it different from polyploidy?
Good distinction. Polyploidy is having extra complete sets chromosomes, like 69 instead of 46 triploidy or 92 tetraploidy. That's usually incompatible with life, lethal very early.
Okay.
Aneuploidy is different. It's a change in the number of a single chromosome. One extra is a trisomy total 47. One missing is a monosomy total 45.
Chromosomy, monosomy, got it. And the sources emphasize that messing with the autosomes chromosomes 1 to 22 is generally much worse than issues with the sex chromosomes X and Y.
That's a really important rule of thumb, yeah. Autosomal aneuploidies tend to have far more severe consequences.
Okay, let's make this concrete. The sources mention a case, Noah, born with some specific signs.
Right, the clinician notes generalized hypotonia, sort of floppy muscle tone, maybe mild feeding issues, upslanted eyes, a flatter facial profile, perhaps a protruding tongue, and that sandal gap.
Sandal gap.
Yeah, a noticeable space between the big toe and the second toe. When you see a constellation of features like that, features that aren't typical, it flags a potential underlying issue.
Like a chromosomal anomaly.
Exactly. So you order the test, the karyotype,
And the result comes back, 47XY plus 21.
and that locks in the diagnosis. Trisomy 21, an extra copy of chromosome 21, Down syndrome.
It's the most common autosomal aneuploidy we see in live births, about 1 in 800 kids across all ethnic groups.
And that extra chromosome 21, it throws everything off balance, doesn't it? Gene dosage.
Precisely. Every gene on chromosome 21 is present in 3 copies instead of 2. So they're expressed at potentially 150% of the normal level. That disrupts complex developmental pathways leading to the characteristic features. And as we said, about 90% of the time, it traces back to that non-disjunction error in maternal meiosis-I. Okay, Down syndrome is the most common. What about others mentioned? Trisomy 18.
Trisomy 18, Edwards syndrome. Much rarer, about 1 in 6,000 live births, and much more severe, illustrating that rule about autosomes. You see features like clenched hands, offer the second finger over the third, fifth over the fourth, rocker bottom feet.
Rocker bottom feet.
Yeah, the sole of the foot is sort of rounded. Survival is very poor. The vast majority, maybe 95%, of trisomy 18 conceptions actually result in spontaneous abortion. The genetic imbalance is just too great.
Wow. And trisomy 13.
Pat's syndrome, even rarer, maybe 1 in 15,000 or so.
Hmm.
Often associated with severe midline defects, problems with eye development, cleft lip or palate, extra fingers or toes, polydactyly. Sometimes areas where the skin is missing on the scalp, called cutis aplasia. Again, devastating consequences.
It really highlights how critical the right amount of genetic material is, especially for those larger autosomes.
Absolutely. Which makes the contrast with sex chromosome aneuploidies quite striking.
Because they're generally less severe.
Much less severe, usually. Partly because of X inactivation. In females, XX, one X chromosome is largely shut down early in development, forming a bar body. This helps balance the dose of X-linked genes compared to males, XY. So having an extra or missing sex chromosome is often better tolerated.
Okay, like monosomy X Turner syndrome.
Right, 45X. Instead of X or XY, just one X. These individuals are female, typically have short stature, maybe a webbed neck, and are usually infertile because their ovaries don't develop properly. It's still serious. In fact, 99% of 45X conceptions are lost spontaneously. But those who are born can live relatively long lives.
Another common one, 47,000 XXY.
Klinefelter syndrome affects about 1 in 1,000 male births, an extra X chromosome. Often they aren't even diagnosed until puberty or later. They tend to be tall and thin, have underdeveloped tests, hypogonadism, and are usually infertile. But again, the overall health impact is generally much less severe than having an extra autosome like 18 or 13.
It's fascinating how the type of chromosome involved makes such a huge difference. So to pin down these diagnoses, to get from floppy baby to 47XY plus 21, you need that picture, the karyotype.
The karyotype is the cornerstone. It's literally an organized picture of a person's chromosomes.
How do they get that picture?
You usually take cells, often blood cells, culture them, stimulate them to divide, and then arrest them right in the middle of mitosis, specifically in metaphase. That's when chromosomes are most condensed and visible.
Okay.
Then you treat them with a stain, typically gene after some pretreatment that's called G-banding. This creates a unique pattern of light and dark bands along each chromosome, like a barcode.
A barcode for each chromosome.
Exactly. That banding pattern lets you identify chromosome 1, chromosome 2, all the way to 22 plus X and Y. Then you arrange them digitally or physically, ordering them by size, from largest to smallest, and by the position of their centromere.
Right, the centromere, that constricted part we see. It divides the chromosome into arms.
Yep. The shorter arm is called the P arm, P for petite. The longer arm is the Q arm, Q, because it comes after P. And at the very ends, you have telomeres, protective caps.
And the position of that centromere matters for classifying them.
It does. Metacentric means it's roughly in the middle, like chromosome 1. Submetacentric means it's off-center, like chromosome 2 or X. But the ones we really need to focus on, clinically speaking, are the acrocentric chromosomes.
Acrocentric, which ones are those?
Chromosomes 13, 14, 15, 21, and 22, plus the Y chromosome, though it's a bit different. For 13, 14, 15, 21, 22, the centromere is way up near one end.
So they have a really short P arm.
Tiny P arm, almost non-existent. And what's on those tiny P arms are basically stalks and satellites containing genes for ribosomal RNA. Now, we have lots of copies of these RNA genes elsewhere, meaning the cell can actually tolerate losing those P arms without much, with any, clinical consequence. And that structural feature makes these acrocentric chromosomes prone to a specific type of structural rearrangement.
Okay, so their structure makes them vulnerable, which leads us beyond just wrong numbers.
Into structural aberrations, rearrangements of the chromosome segments themselves.
Like what?
Things like deletions losing a piece, duplications gaining an extra piece, inversions, a segment gets flipped around, and translocations pieces get swapped between different chromosomes.
And these can be balanced or unbalanced?
Right. Balanced means all the genetic material is still there, just rearranged. Often the person is fine, but they might have problems having children. Unbalanced means there's a net loss or gain of genetic material. That usually causes problems.
Let's take deletion as an example. Losing a piece. Why is that often so bad? You mentioned haploinsufficiency.
Haploinsufficiency, great term. Most of our genes work best when we have two working copies, one from each parent. They produce a certain amount of protein needed for a cell to function correctly.
Okay.
If you delete a segment of a chromosome, you lose one copy of all the genes in that segment. And for those genes, you only have one working copy left.
You're only making 50% of the protein.
Roughly, yeah. And for many developmental genes, 50% just isn't enough. That's haploinsufficiency. The single remaining copy is insufficient to do the job properly, and that leads to the clinical features of deletion syndromes.
Like the example in the sources, Cri-du-Chat syndrome.
Classic example. Cri-du-Chat means cry of the cat in French, named because infants with this often have a high-pitched cry that sounds like a kitten. It's caused by a deletion on the short arm of chromosome 5.
So missing a piece of 5P.
Right, and the severity of the developmental delay, the intellectual disability, the characteristic cry, it all stems from haploinsufficiency for the genes lost in that deleted segment.
Okay, so whether it's an extra chromosome, a missing one, or a missing piece, we need a way to describe these findings precisely, universally.
Absolutely critical. Imagine trying to compare research or clinical cases from different countries if everyone used a different naming system. It would be chaos.
So we have a standard.
We do. The International System for Human Cytogenomic Nomenclature, ISCN. The latest version is ISCN 2020. It's the global standard.
Yeah. ISCN. How does it work? Can we decode it a bit?
Sure. It's built on precision. First, locating something on a chromosome. A designation like 7q31.2 tells you exactly where to look.
Okay, break that down. 7q31.2.
Chromosome 7, long-armed Q, region 3, band 1 within that region, and subband 2 within that band. It's like a very precise genetic address.
Wow, okay. And the overall format for describing a karyotype result?
It's pretty logical. You start with the total number of chromosomes, then a comma, then the sex chromosomes, then another comma, followed by any abnormalities.
So normal female,
46XX,
normal male,
46XY,
the boy with Down syndrome we discussed,
47XY plus 21. The plus 21 indicates an extra chromosome 21.
Xander syndrome,
45X just lists the single X present.
and for structural things like Cri-du-Chat.
You use abbreviations, del for deletion, dup for duplication, in for inversion, T for translocation. So Cri-du-Chat could be written as 46XX del 5P, or more precisely, sometimes you'll see the specific breakpoints, like 46XX del 5P13, indicating the deletion starts at band P13 on chromosome 5.
Got it. And sometimes people have a mix of cells, Mosaicism.
Yeah, mosaicism is when someone has two or more genetically distinct cell lines originating from a single fertilized egg.
Huh.
ISCN handles that too. You use most at the beginning, then list the cell line separated by a slash, often with the number of cells observed in brackets. Like most 45,000 x 46,000 XX20 means mosaic Turner syndrome, 30 cells were 45X, and 20 cells were 46XX.
It's incredibly systematic, a true universal language for genetics.
It has to be. It allows clinicians and researchers worldwide to communicate complex findings without ambiguity.
So looking back, we've gone from the sheer elegance and complexity of meiosis, designed to generate millions of unique combinations,
To the specific points where it can fail non-disjunction, leading to conditions like trisomy 21, trisomy 18, Turner syndrome.
and we've seen how we map these changes visually with karyotypes and describe them precisely using ISCN, from the biology of swapping DNA to the code 47XY plus 18.
Exactly. That ability to distill complex biology into a standardized notation like 47XY plus 18 for Edwards syndrome or 46XX Delphi 5P for Credusha
Yeah.
is what transforms this knowledge into a powerful clinical tool.
Fantastic. Thanks for walking us through these sources. It's been a real deep dive into the mechanics and language of our chromosomes.
My pleasure.
It leaves us with a final thought, circling back to the start. We said meiosis aims for diversity, and crossing over actually helps reduce errors, yet errors still happen, especially with maternal age. So the provocative question for you to ponder is, what does this persistent rate of meiotic error tell us about the fundamental biological trade-off? The constant tension between maximizing genetic variety for the species and the absolute need for genomic stability within the individual. Something to think about.